Welcome back to the deep dive. Today we are taking a massive leap into the foundational machinery of the human body. We're talking about a scientific breakthrough that has dramatically redefined and maybe almost quintupled our map of the human interactome.
That's exactly right. We are deep diving into a really fascinating new study. It systematically mapped the critical connections that, allow human life to function. Protein interactions, or PPIs.
PPIs, okay, so these are like the molecular handshakes inside our cells.
Precisely. Think of them as the essential gears and levers. They drive virtually every biological function. Everything from fighting off a virus to metabolizing your lunch.
The scale of what they did here sounds incredible. They screened 200 million possible protein pairs and predicted nearly 18,000 interactions.
That's the number, yeah. 17,849 high-confidence predictions. It's a huge step forward.
But, okay, here's the obvious question then. Why now? Why haven't we had this kind of map before? It seems pretty fundamental.
Well, it boils down to a couple of big problems. Data inconsistency and, frankly, technical limitations. The traditional experimental methods we used, things like yeast-2 hybrid or mass spectrometry approaches, they're powerful, sure.
But not perfect.
Not even close in some ways. They often test proteins outside their natural environment, not under physiological conditions. And that leads to incredibly high rates of both false positives, seeing interactions that aren't real, and false negatives, missing ones that are.
So the existing data was just messy.
Very messy. The source material really highlights this huge gap. Experts estimate the real human interactome, the total web of connections, is probably somewhere between 74,000 and maybe 200,000 PPIs.
Okay, that's the estimate. But what do the databases actually say?
Well, that's where it gets wild. If you just count up everything reported in the major public databases, UniProt, BioGrid, String, you get over 1 million reported interactions.
1 million? Compared to maybe 200,000 real ones? That's a lot of noise.
Exactly. And it's not just the sheer number, it's the lack of agreement. When you apply a strict filter, say, only counting interactions confirmed by at least 3 independent experiments, what we'd call high-confidence data,
the number must drop dramatically.
it plummets down to just 3,988, fewer than 4,000 reliably confirmed interactions defining the core machinery of human life.
Wow. Okay, so that data deficit makes it crystal clear why we needed a different approach.
Yeah.
An unbiased computational one.
Absolutely necessary.
Cool.
And that brings us to the mission of this study. Conduct a comprehensive proteome-wide screen. But to do that, you couldn't just use the old tools or the messy data. They needed 2 massive innovations.
Right. You mentioned harnessing petabytes of ignored data and a faster deep learning model. Let's unpack that first one. How did they make the input data? What was it? 7 times deeper?
Yeah, 7 times deeper. It starts with the basis of how you computationally predict these interactions. Coevolutionary analysis.
Coevolution. Like 2 things evolving in response to each other.
Exactly. Imagine the classic lock-and-key idea for proteins that need to fit together. If a mutation happens that changes the shape of the lock protein right at the interaction point.
Then the key protein needs to change too to maintain the fit.
Precisely. If that interaction is important for survival, evolution will favor a compensatory mutation in the partner protein, a co-mutation. Over millions of years across many species, you get this subtle pattern of linked changes at the interaction interface.
So tracking those linked changes across species tells you which proteins likely interact.
That's the core idea. But to detect that faint coevolutionary signal, you need incredibly deep arable, multiple sequence alignments, or MSAs. An MSA lines up the sequences of the same protein, the orthologs from thousands of different species. It's like a deep evolutionary history book for that protein pair.
And the problem for humans was we didn't have enough related species with good genomes sequenced. The history book was too thin.
Exactly. We had a limited number of high-quality, fully assembled animal genomes. The data was just too shallow. The statistical signal for coevolution was weak.
Okay, so how did these researchers solve that? You said they used ignored data.
They did something really clever. They basically stopped waiting for perfectly assembled genomes. They went straight to the raw ingredients. 30 petabytes of raw, unassembled genomic data.
30 petabytes, that's 30 million gigabytes, right? An astronomical amount of raw sequence reads.
It's immense. They mined this from public databases like the NCBI Sequence Read Archive places, where researchers dump raw sequencing output. It's like instead of reading published books, they went to the printing press floor and gathered up all the snippets and fragments of text.
And somehow assembled protein sequences from that raw mess.
They developed a specialized computational pipeline to do just that. It fished out and assembled protein coding sequences directly from those raw reads. They focus on getting data from over 21,000 diverse eukaryotes, especially chordates and arthropods, to get that deep evolutionary comparison.
That sounds incredibly complex, especially with eukaryotic genes having introns and exons.
Oh, it was a huge bioinformatics challenge. But the payoff was enormous. By building their own protein sequence database this way, they created these custom alignments they called omicMSAs.
omicMSAs. Got it.
And here's the key metric we mentioned. These omicMSAs were, on average, 7-fold deeper than the standard alignments you'd get from established databases like UNIREF 100.
7 times more evolutionary history to look at for those co-mutation signals.
Exactly. It dramatically amplified the coevolutionary signal. This wasn't just tweaking a model. They fundamentally improved the quality and depth of the input data the model could learn from. This deep data was essential for everything that followed.
So better data was the first pillar. That makes sense.
Yeah.
Okay, let's pause for a quick recap. We're diving into a study that massively expanded our map of human protein interactions. We started with the problem. Existing databases were messy, inconsistent. Maybe only 4,000 interactions were truly high confidence.
Right. And the reason computational methods struggled before was often due to shallow evolutionary data, especially for humans.
So the first big innovation was creating omicMSAs. They tackled the data problem head-on.
Yep, by mining 30 petabytes of raw, unassembled genomic sequences
From over 20,000 species.
to build alignments that were 7 times deeper, boosting that crucial coevolutionary signal.
Okay, so now they have this amazing, super deep data set. Best data ever for this task. But the second problem was scale, right? Screening 200 million potential pairs. Even with great data, how do you make the machine learning fast enough? Existing tools like AlphaFold 2 weren't up to it.
Exactly. AlphaFold 2, or AF2, is revolutionary, no doubt. Amazing for predicting the 3D structure of a single protein, or maybe a known complex. But running it on 200 million potential pairs is just far too slow, computationally prohibitive. You'd need decades of computing time.
So they needed speed, but without losing the accuracy that deep learning like AF2 provides.
That was the goal. And that led to the second major innovation, a new optimized deep learning network called Rose-de-Fold-2 PPI, or RF2 PPI.
Okay, based on the name, it's related to Rose-de-Fold, another structure prediction tool.
Yes, they adapted the Rosetta-Fold architecture specifically for predicting interactions, and crucially, they engineered it for speed. RF2 PPI turned out to be approximately 20 times faster than using AF2 for the same interaction prediction task.
20 times faster. Okay, that makes the proteome-wide scale seem actually feasible. But speed isn't everything. How did they train it effectively? You said the known high-confidence PPI data is tiny, those 4,000 examples. That's not much for training a deep learning model.
It's definitely not enough on its own. And this is where I think the most ingenious part of the study comes in. They used a strategy called training augmentation using domain interactions, DDIs.
Okay, break that down. DDIs.
So they started with a small set of known PPI structures from the Protein Data Bank, PDB. That gave them about 13,000 examples after clustering. Still small. Then they made a really clever hypothesis.
Which was?
That the way functional domains interact within a single, large, folded protein, the interfaces where 2 distinct parts of the same protein chain touch, that those interfaces structurally resemble the interfaces where 2 completely separate proteins come together to interact.
Ah, I see. So the physical rules, the geometry, the kinds of amino acid contacts involved in making 2 parts of one protein stick together are similar to how 2 different proteins stick together.
Precisely. Domains are like reusable building blocks in proteins.
Yeah.
It makes sense they'd use similar connection rules, whether they're part of the same chain or different chains. So they leveraged this idea. They went to the huge AlphaFold protein structure database, which contains predicted structures for millions of proteins.
Including those 200 million monomer structures they mentioned.
Exactly. And from those structures, they computationally identified and extracted instances where domains were predicted to interact within those single proteins. They harvested a massive distillation set of these DDIs.
How massive?
They got over 223,000 high-quality DDI examples after clustering. Compare that to the 13,000 PPI examples they started with.
Wow, that's a 17-fold increase in training data, just by using interactions within proteins as proxies for interactions between proteins.
Right. It gave the model so much more structural information to learn from, based on reliable predictions from AF2 itself, essentially. And this strategy, combining the deep omicMSA coevolution data with this massive DDI structural data, gave RF2 PPI a really interesting advantage.
Which
It
was?
turned out to be particularly good at predicting weak and transient interactions.
The really tricky ones, the ones that happen quickly and maybe don't involve large contact surfaces.
Exactly. Those are often missed by experimental methods and other computational models. They typically have smaller interfaces.
Why would RF2 PPI be better at those? Especially compared to something super accurate like AF2, which you'd think would nail the structure.
It seems to be about what the model prioritizes. AF2 is heavily focused on achieving the best possible geometric fit, the most stable final 3D structure. RF2 PPI, perhaps because of its training on omicMSAs and DDIs, seems to put more weight on the coevolutionary evidence. Is there strong evolutionary pressure suggesting these two must interact, even if the predicted structural interface isn't huge or perfectly rigid?
So for those weaker interactions, the evolutionary smoking gun might be clearer than the structural footprint.
That seems to be the case. RF2 PPI could pick up on that coevolutionary signal more strongly, giving it an edge for these faint, fleeting, but often biologically critical interactions. That's a huge win.
Okay, so we have the deep data, omicMSAs, and the fast, well-trained model, RF2 PPI. But they didn't just throw 200 million pairs into RF2 PPI, did they? You mentioned efficiency.
No, that would still be too much, even at 20x speed. They designed a very clever multi-stage screening pipeline, a funnel, basically.
Smart. What were the stages?
Stage 1 was a simple biological filter, localization. Do the two proteins even exist in the same part of the cell? If one protein is only ever found in the nucleus and the other is always stuck in the cell membrane, they're very unlikely to interact directly.
Makes sense. That must have cut down the numbers significantly.
It did. It immediately reduced the 200 million potential pairs down to about 111 million pairs, where the proteins share the same cellular compartment or their locations are unknown. So roughly half the problem size.
Okay, 111 million still sounds like a lot. What was filter number 2?
Stage 2 used a much faster, simpler computational method called direct coupling analysis, or DCA.
DCA. How does that compare to RF2 PPI?
DCA is about 5 times faster than RF2 PPI because it's not a deep learning model. It's more of a statistical analysis that directly looks for correlated mutations, those coevolutionary signals, but in a less sophisticated way than the full model. Think of it as a quick pre-scan. If the DCA score was low, suggesting weak coevolution, they basically threw that pair out.
So a quick check to weed out the clearly non-interacting pairs.
Exactly. This narrowed the field from 111 million down to about 43.6 million pairs that showed at least some promising coevolutionary signal.
Getting more manageable. So these 43 million pairs were the ones fed into the RF2 PPI model.
Yes. Stage 3 was the main RF2 PPI screen. It assessed those 43.6 million pairs and assigned an interaction probability. They kept pairs with a probability above a certain threshold, RFI prob 0.3. This yielded about 1.9 million top candidate interactions.
Okay, nearly 2 million. Getting closer to the final number. What was the last step?
Stage 4 was the final quality check, bringing back the slower but highest accuracy model, ElePhold 2. They used AF2 to generate detailed 3D models for those final 1.9 million candidate pairs and calculate a final high-confidence interaction probability, AFM-prob.
So using the fastest tools for broad screening, then reserving the most powerful tool for the most promising candidates.
Precisely. It's a tiered strategy maximizing both scale and final accuracy. This funnel approach is what allowed them to arrive at the final set of 17,849 predicted PPIs with that estimated 90% precision.
Incredible. So 17,849 high-confidence predictions. How does that number change the landscape of the known human interactome?
It changes it significantly. Remember that baseline of only 4,000 interactions agreed upon by multiple experimental methods. This work adds nearly 18,000 computationally derived high-confidence interactions, many with predicted 3D structures. And crucially, 3,631 of these predictions, about 20% of their final set, were completely novel. They weren't found in any of the major experimental PPI databases like Uniprot, BioGrid, or String.
20% are brand new high-confidence interaction predictions. That's huge.
It really is. And they showed strong supporting evidence. For instance, the pairs they predicted were much more likely to share the same biological function annotations and be found in the same cellular quotients compared to randomly chosen protein pairs, or even pairs from less stringent experimental datasets.
The biological context matches up, adding confidence.
Absolutely. It suggests these aren't just random computational guesses. They make biological sense.
Okay, let's talk function. One area where experimental methods really struggle is with transmembrane proteins, TMPs, the ones embedded in cell membranes. They're greasy, hard to isolate. How did this computational pipeline fare with those?
That was actually a notable success highlighted in the study. Because the method relies heavily on sequence and coevolution, it doesn't have the same difficulties with membrane proteins that experimental techniques do. They found that their set of predicted PPIs actually contained a higher fraction of interactions involving transmembrane helis, TMHs, compared to experimentally determined structures in the PDB.
So this approach might be particularly powerful for mapping interactions in that dark proteome within the cell membrane.
It certainly looks that way. It's opening up visibility into interactions that were very difficult to study before.
Let's make this concrete. The study presented some specific case studies linking these predictions directly to human diseases. Can you walk us through one or two?
Sure. The first one connects to a condition called ocular albinism type 1, which affects vision and pigmentation. The pipeline predicted a novel interaction between a protein called GPR143, which is an unusual type of receptor found in pigment cells, and another protein called HPS1 involved in making the structures that hold pigment, melanosomes.
Okay, a predicted link between two proteins involved in pigmentation. Did it connect to the disease?
Directly. When they mapped known mutations that cause ocular albinism type 1 onto the predicted structure of this GPR143 HPS1 interaction, they found that 4 different disease-causing mutations, single amino acid variations, or SAVs, landed right at the predicted interface where these two proteins touch.
Wow, so it strongly suggests the disease isn't just caused by GPR143 being broken, but specifically by its inability to interact correctly with HPS1.
That's the implication. Disrupting this specific molecular handshake seems to be the pathogenic mechanism. That gives you a very precise target if you wanted to design a therapy.
Incredible molecular detail. What about an example related to the immune system?
Yeah, another compelling case involved immunity. They predicted an interaction between an inhibitory receptor called KLRG1, found on immune cells like natural killer cells and T cells, and TLR3. TLR3 is a key sensor. It detects double-stranded RNA, which is often a sign of viral infection.
Okay, so an inhibitory receptor potentially interacting with a viral sensor. What's the disease link?
They looked at mutations associated with a rare but devastating inherited immunodeficiency disorder. This disorder makes people highly susceptible to herpes simplex virus 1 encephalitis, a severe brain infection. And again, they found that mutations causing this susceptibility were located right at the predicted KLRG1-TLR3 interaction interface.
So if KLRG1 normally puts the brakes on TLR3 signaling, and that interaction is broken by a mutation,
you might get an overactive or uncontrolled TLR3 response when the virus appears, leading to damaging inflammation or some other dysfunction that causes the severe disease. Again, it provides a potential molecular explanation for the disease pathology.
these examples really show the power of having that structural context for interactions. Did they also find insights into larger protein complexes?
Yes, the predictions weren't just pairwise. They could also help assemble larger molecular machines. For example, they looked at the tubulin polyglutamylase, TPG complex, which modifies microtubules in the cell. Their predictions suggested 3 additional subunits belonged to this complex that weren't previously confirmed: TBC1D19, CSTP-BELO, and SNBR.
Adding new parts to the machine's blueprint.
Exactly, and specifically for TBC1D19, the predicted structure showed it interacting with 5 other subunits in the complex. But intriguingly, its binding site was located away from the main catalytic or active site of the enzyme.
Suggesting it might regulate the enzyme's activity indirectly, like allosteric regulation.
That's the idea. Binding at a distant site could cause a shape change that affects the enzyme's activity, providing a more subtle level of control. It gives clues about sophisticated regulation within these complex machines.
Okay, so let's pull this all together. The 2 huge technical leaps were, first, creating the ultra-deep omicMSAs by mining petabytes of raw sequence data to boost the coevolutionary signal.
Right, solving the data depth problem.
And second, developing the efficient RF2 PPI deep learning network, cleverly trained using that massive data set of domain interactions harvested from AlphaFold structures.
Solving the speed and training data problem.
And the result is this data set of nearly 18,000 high-confidence PPIs, nearly tripling the number with good 3D structural models available.
And providing immediate biological insights. They found that over 15% of known disease-associated mutations map onto these newly predicted interaction interfaces. The potential impact on understanding disease is immediate.
It's truly remarkable progress, but there's always a what's next. You mentioned earlier they estimated their predictions cover maybe 8 to 22% of the total human interactome. That's a huge jump, but still leaves a lot unknown. What's the big remaining challenge?
The major frontier now is accurately predicting the interactions involving the most flexible parts of proteins, particularly those mediated by intrinsically disordered regions, or IDRs.
IDRs, can you remind us what those are?
Sure. If you think of most well-studied proteins or protein domains as having a relatively stable, fixed 3D shape, like intricate Lego blocks, IDRs are different. They're segments of proteins that don't really fold into a stable structure on their own. They're more like flexible strings or ropes.
Okay, floppy bits of protein.
Exactly. They often only take on a more defined, albeit still potentially dynamic, structure when they actually bind to a partner protein. Because they're inherently flexible and often interact transiently using small surface areas, they are incredibly difficult targets.
Difficult for experiments to catch, and difficult for computational models that are often trained on more rigid structures.
Yeah, precisely. Both experimental methods struggle to capture these fleeting interactions, and computational models like AF2 or even RF2 PPI have a harder time when there isn't a clear, stable interface driven by strong coevolutionary geometry. Predicting how these molecular ropes tie things together is the next big hurdle.
That definitely sounds like the challenge for the next deep dive in this field. So, wrapping up, let's leave our listeners with a final thought. What does this massive expansion of the Interactome map mean for you, personally? Consider a complex disease you might know about, maybe cancer, Alzheimer's, autoimmune disorders. We know they involve protein dysfunction. Now that we can suddenly see thousands of previously unknown high-confidence connections between proteins, complete with 3D models, how quickly might this change the game for developing new therapies? Could the focus shift from just targeting a single broken protein to instead finding ways to mend or modulate the specific interactions, those crucial molecular handshakes that have gone wrong? That seems like a really profound shift in perspective.